Hi everyone, my name is Suali from the University of Illinois, and we are going to discuss how the energy and nutrient digestibility are changed when dietary fat increases in diet fed to growing pigs. This work was presented in 2025 ISEP meeting. Equipment to determine net energy values of feed ingredient is available only at a few universities in the world. But the University of Illinois recently constructed such a facility. This facility has a capacity to hold the multiple number of pigs depending on the size and is installed with indirect calorimeter chambers that are airtight, which enables us to measure the gas consumptions and production of pigs. In addition, because pigs are group housed, pigs share one feeder and move around, which may impact the energy expenditure. As you can see here in the cartoon, the feces and urine pens are installed inside the chamber, which enables us to collect feces and urine separately. Therefore, using the system, concentrations of digestible energy, metabolizable energy, and net energy, and digestibility and retention of a nutrient can be determined in diet fed to pigs on an ad libitum basis. And because the way pigs are housed is similar to the way pigs are housed in commercial settings, this system is believed to generate values that are representative of commercial settings. Using this system, accurate prediction of net energy in diet fed to group housed pigs requires an understanding how each nutrient class influences energy utilization and nutrient digestibility. Therefore, we have conducted a series of experiments to determine net energy and nutrient digestibility in diets containing a wide range of carbohydrate, lipid, and protein to develop a new NE system ultimately. Among the energy containing nutrients, let's talk about fat this time. Lipids are highly digestible and metabolizable in the body, which provide greater energy values in fat than protein or carbohydrates. There were no data how increasing soybean oil changes the digestibility of nutrients and the energy in diet fed to group housed pigs. Therefore, an experiment was conducted to test the hypothesis that increasing dietary fat increase net energy and nitrogen balance and the digestibility of nutrients. There were 6 experimental diets. The first diet only contained corn and soybean meal and contained no soybean oil. The next 5 diets contained corn, soybean meal, and soybean meal from 1.5, 3, 4.5, 6, and 7.5%. More crystalline amino acids were added to the 5 diets to maintain amino acid to metabolizable energy ratios. It was expected that concentrations of fat increased in these diets. Indeed, the analyzed acid hydrolyzed ether extract was low in the first diet and increased up to 10.45% in the last diet. We utilized 2 groups of 24 pigs and 4 pigs were housed in a chamber. In each group, the 6 chambers were allotted to 6 treatments using a 6x3 incomplete Latin square design. Because there were 2 groups, there were a total of 6 replicates per treatment. Pigs had ad libitum access to feed and free access to water. After 7 days of adaptation, feces and urine were collected for 6 days using the time-to-time procedure. During the collection period, consumption of oxygen and productions of carbon dioxide and methane were also measured to determine the total heat production. Diet and fecal samples were analyzed for dry matter, gross energy, nitrogen, and ash. Urine samples were analyzed for gross energy and nitrogen. Using the analyzed values and weights of feces and urine, the apparent thoracic digestibility of nutrients and energy and retention of nitrogen were calculated. Concentrations of digestible, metabolizable, and the energy were also calculated. The statistical model included diet as a fixed variable and group, period, and timbre as random variables. Contrast coefficients were used to determine linear and quadratic effects of increasing dietary fat. Significance was set at alpha less than 0.05 and tendency at zero point 0.1. Moving to the result, let me set up the slide first. To better communicate, the inclusion rate of soybean oil was used on the bottom, which ranged from 0 all the way up to 7.5%. The results indicated that the apparent tortuous digestibility of a dry matter and the ATDD of gross energy decreased and increased by increasing soybean oil in diets. And now we are looking at nitrogen balance data, and the ATDD of nitrogen was not affected by increasing soybean oil in diets. However, even though I am not showing you the urine data, daily urine nitrogen excretion decreased, which increased retention of nitrogen as soybean oil increased in diets. However, the ATDD of ash was not influenced by dietary fat. We were able to calculate RQ, which is the ratio between carbon dioxide production to oxygen consumption, and the result indicated that values for RQ during the collection period, that is a fat state, tended to decrease. This was expected because fat oxidation is known to consume more oxygen for each unit of carbon dioxide produced, which reduced the RQ compared with protein or starch from corn and soybean meal. Concentration of net energy increased as soybean oil increased in diets. The increase in energy can mainly be explained by the fact that lipids generally contain more energy than other nutrients, but also the presence of dietary oil increases retention time and thus increases other nutrients digestibility. We also conducted another experiment to determine the apparent ileal digestibility of a dry matter, starch, and amino acids to demonstrate the relationship between net energy and nutrient digestibility in the same diet. Data indicated that the ileal digestibility of a dry matter was not affected by dietary fat, but the starch digestibility was increased by increasing soybean oil in diets. The ileal digestibility of acid hydrolyzed ethyl extract and amino acid was also increased by increasing soybean oil, which demonstrated that the increases in the net energy were also from the increases in the starch and amino acid digestibility, which may increase nitrogen retention that we have discussed in the previous slide. In conclusion, increasing dietary soybean oil increased energy digestibility and concentration of net energy in diets, and decreased the ratio between produced carbon dioxide and consumed oxygen. The linear increase in nitrogen retention indicated that more body protein was synthesized as increased dietary soybean meal made more energy available for the pigs. Thank you for listening to my presentation, and I would like to acknowledge everyone from Dr. Stein's lab. If you want to learn more about our research that are conducted in the Stein Monogastric Nutrition Laboratory, please visit our website. Thank you.